Okay, enough of that. So it's my pleasure to introduce Devon DeRaad. Um, it's really been a pleasure to have him in the lab for the last several years. He arrived from doing a bachelor's degree at Occidental and really hit the ground running here in our lab. And he sort of combines a great set of attributes for a graduate student and an academic, which is a great curiosity about the natural world, ability to formulate really good scientific questions, and incredibly hardworking. And so he's able to follow through on answering those questions and publishing the papers and submitting the revisions and the next round of revisions and everything else and just getting things done. So he's been incredibly successful. Um, as a measure of his success, by my count, he's been a co-author on 22 papers since he was here at KU and a lead author on 9 from a variety of journals: systematic biology, evolution, molecular ecology. Just kind of runs the gamut of kind of the journals that you want to be publishing in if you're in evolutionary biology, especially in systematics. He has also raised over $10,000 through small grants to fund his research, and he was supported by a self-graduate fellowship when he was here. And he's really kind of taken advantage of all the opportunities given to him, allowed to him, here at KU, and really made the most of it. So incredible productivity, no doubt, but also he's tremendously selfless with his time as well. He's helped out so many people with data analyses, things like that, whether it be through like formal classes, like doing the RAD-seq working group, which he already knew how to do it all, no problem, but he set up times to teach everybody else how to do it. To just informal things, the number of people I saw popping in to pick his brain about why they kept getting error messages out of some program, is pretty incredible. So needless to say, I tried to keep him around as long as possible, but you can only do that for so long. And now, starting in August or the fall, he's received a postdoctoral fellowship from UCLA to continue studies of evolutionary genomics of birds. So, Devon?
Okay. Is the mic working? Can you hear me? Okay, great. Thanks, Rob, for that great introduction. And I'm really excited to be here talking to you today about my dissertation, which is titled Updating the Tree of Life, Case Studies in Birds of the Pacific Islands. So this is a quick outline of the talk. I'm going to give you some background on the motivation behind my dissertation, and then I'll go through each of the 3 chapters, which focus on 3 different groups of birds from the Pacific Islands, and end with some broadly relevant conclusions. But I want to start with this quote, which says, "Nothing in biology makes sense except in the light of evolution." And I like this quote because I think understanding evolution gives us a lens that can make even the messiest patterns in nature start to make sense if we dig deep enough. And I think most of us who have dedicated our careers to studying evolution probably agree with this quote, and would probably also agree that the most important model ever developed for understanding evolution is the phylogeny, or phylogenetic tree. And this is the original phylogeny sketched in Darwin's notebook about 150 years ago. And you can see written next to it in large letters the words "I think," which is honestly one of my favorite things ever, because it lets us know that all evolutionary biologists have had imposter syndrome since literally the moment the field was founded. But in all seriousness, Darwin was totally correct in his thinking that all of life on Earth was related evolutionarily and actually radiated out into the incredible diversity we see today from a single common origin. And studying these patterns has become known as the field of phylogenetics. And at its simplest level, phylogenetics just means reconstructing phylogenies, or these tree-like diagrams that connect lineages via common shared ancestry through time. And on these plots, the more recently connected 2 lineages are, the more closely related they are. So here, A and B are more closely related to each other than either is to C. And one of the terms I'm going to be using repeatedly today is bifurcating, which just means that one branch in the tree always splits into two, which is the simplest type of phylogenetic tree. In contrast with more complex trees, they could have multifurcations where 3 or more branches emerge simultaneously, or complex network edges connecting the branches in additional non-bifurcating ways. But regardless of the details, the practice of building these tree-like diagrams to understand evolutionary relatedness, aka phylogenetics, is now ubiquitous in modern science. From forensics for what we would call DNA fingerprinting, which is routinely used to identify whether DNA recovered from a crime scene belongs to a given individual, to medicine, and most famously in recent years for identifying specific types of viruses during the COVID-19 pandemic, where researchers used phylogenetic trees to classify COVID-19 strains into evolutionary groups, which helped us understand how the virus was evolving and how we could counter it in real time. But today I'm going to be focusing mainly on using phylogenetics as an approach for classifying organisms and answering biological questions. And there's a long history of this type of phylogenetics work in museums across the globe, specifically here at the KU Natural History Museum, where I worked as a PhD student. And from the very beginning of this museum-based systematics work, researchers built these phylogenies for specific groups they were interested in, like these butterflies. But almost immediately, there became a specific interest in not just reconstructing small phylogenies, but also in constructing a single coherent tree of life, like the ones drawn here. And this became something of a holy grail in evolutionary biology, the notion that we could construct a single tree encompassing everything from bacteria to mammals that would describe the history of all life on Earth. But at the time, the only way to construct these trees was to carefully study physical museum specimens. But in the years since, as you might have guessed, we have updated our approach to generating phylogenetic trees, and that's because we now have the power of DNA sequencing. And not just a little DNA sequencing, but tons of DNA sequencing. And that's exemplified by this graph, which shows the cost of sequencing a single human genome, which has fallen from around $100 million in the year 2000 to just a few $100s today. So for me, someone coming along in my academic career at this exciting time, following this massive drop in the cost of DNA sequencing, I really wanted to combine the exceptional sequencing power of the genomic era with the incredible treasure trove of biodiversity that's available at the KU Natural History Museum, which can be thought of as a library of biodiversity with archives holding the genetic code of millions of organisms. And in particular, because of years of dedicated field collecting efforts, we have an incredibly extensive tissue collection of birds of the Pacific Islands. And in working with this exceptional collection, previous students from the Moyle Lab have published a swath of phylogenetic studies of the birds of the Pacific Islands. And these studies have repeatedly validated the importance of studying the biodiversity of this area of the world, which is relatively remote and poorly known to Western scientists, in order to better understand the process of evolution and the accumulation of biodiversity across the globe. So for my dissertation, I wanted to revisit each of these 3 studies that I'm showing here on the screen, taking advantage of that incredible drop in the cost of genomic sequencing to generate larger genome-scale DNA sequence datasets and see what this increased resolution could reveal about the evolutionary histories of birds of the Pacific. And the reason my title includes Updating the Tree of Life is that I'm going to try to convince you today that these studies are important, not just for understanding the specific histories of these birds, but also for what they can teach us about the general practice of phylogenetics and about the nature of that holy grail of evolutionary biology, the tree of life. So this brings me to chapter 1, where I studied a group of birds known as the Totirampus kingfishers. And I wanted to let you know that this project was published in the journal Molecular Ecology last year, and that's actually my photo of a beach kingfisher from the Solomon Islands that was featured on the cover of the issue. So if you're interested in all the details, I'd highly encourage you to read that paper, and I'm just going to give you some quick highlights today. And to orient you geographically where this project takes place, I want to show this map of the Pacific. And I promise there's not going to be a quiz on all of this, but I want to emphasize that the Pacific Ocean covers a massive proportion of Earth and contains approximately 30,000 islands, each with their own unique biodiversity and cultural identity. And the project I'm talking about today is on the Totirampus kingfishers, which are known for their extraordinarily rapid radiation across essentially this entire region. Now, I've highlighted here with the red outline the general distribution of the 8 species that we focused on for genetic sampling on this particular project. And this set of maps shows the incredible geographic breadth of the samples we were able to secure for this project. And I want to highlight that each of these genetic samples has an associated museum specimen, either here at KU or in another collection that collaborated on this project, allowing us to verify the connection between the genetic data we generate and the phenotypes that we see in nature. So to start, I'm going to give some quick background on the DNA sequence data that I generated for this project. As I mentioned previously, the process of getting DNA sequence data is becoming continuously cheaper and easier, but especially 5 or 6 years ago when we were generating some of the data I'll be talking about today, it still wasn't financially feasible to use the approach I'm showing here, where you actually sequence the entire genome of every individual in your study. So instead, we used a reduced representation approach, where we only sequence DNA from very specific locations throughout the genome, as I've shown in this bottom panel. And this is a very efficient and cost-effective way to get DNA sequence data from thousands of places throughout the genome. But because we're only targeting certain sequences, there are sources of technical error and issues that can arise during this process. And I want to highlight that I actually spent a significant portion of time during my PhD dedicated to understanding these technical challenges, which led me to develop this R package called SNP filter. And the package will help you visualize parameters and perform quality filtering steps to get a set of high-quality single nucleotide polymorphisms, or SNPs, that you feel confident in. And if you work on SNP data, the package is freely available on CRAN, and I encourage you to try it out on your own datasets. So now that I had generated a genomic data set for this group, which amounted to around 2,000 loci, or pieces of the genome, that I could really trust, I wanted to understand how all of the samples we sequenced were related to each other. And to do so, I made a phylogenetic network. And the way to read this network is that, excuse me, the physical distance you trace between samples on the network corresponds to exactly how similar the samples are in DNA sequence data. So for instance, these two samples from within the same species are very closely related because we only have to travel a short distance on the network to connect them. In contrast, if we want to connect samples from these two different species, we have to travel a greater distance through the network. And what this overall pattern told us is that the samples within each species are more closely related genetically to each other than they are to any individuals from other species. And based on that, we can now confidently say that the recognized species in this group, which are circled on the network, accurately describe the major genomic lineages that are present. And we can also say that we don't see any immediate signs of gene flow or hybridization between species in this network because we don't have samples showing up in the middle of the tree where I put those red X's, which would suggest intermediate ancestry. And this is really important finding because if we want to reconstruct the evolutionary history of these birds on a larger scale, we have to be confident that the individuals we're using actually belong to the species that we expect them to. So now that we feel confident in that, we want to use these samples to reconstruct an overall species tree, which summarizes the higher level relationships between the major genomic lineages in the group. And the way this works is that I generated a separate tree for each of the 2,000 chunks in the genome that we sequenced, and I'll refer to these as gene trees. You can see here an example of many gene trees all plotted on top of each other. And then I'll use those gene trees to reconstruct a single summary species tree that describes the genome-wide relatedness between the major lineages in the group. And this is the resulting species tree, which shows a set of evolutionary relationships, and they seem plausible. But as I mentioned in the intro, this project is actually following up on a previous study, Anderson et al. 2015, who sequenced 2 mitochondrial genes from the same set of 8 species, among many others. But I wanted to compare the relationships that I recovered using this genome-wide data to those recovered by Anderson using mitochondrial sequence data. And a quick reminder, in birds, the nuclear genome is about 1 billion base pairs long, and there are 2 copies of every chromosome, and these chromosomes recombine or reshuffle every time they're passed on. Meanwhile, the mitochondrial genome is small, only about 17,000 base pairs, and it's inherited maternally, so it never experiences recombination or reshuffling. And because these 2 parts of the genome have such fundamentally different properties and are inherited differently, we wanted to compare the patterns of relatedness that we got between the 2. And here are those 2 trees lined up next to one another, showing that we recovered different sets of evolutionary relationships between these 2 different types of DNA sequence data. And the term that we use to describe this phenomenon is mitonuclear discordance. And because of the different properties of the mitochondrial and nuclear genomes, it's been repeatedly shown that hybridization and gene flow between species, which I've shown as this red cartoon arrow, will affect these two regions differently, often causing mitonuclear discordance. So I wanted to understand whether gene flow might be the cause of the mitonuclear discordance we're seeing here in kingfishers. To test this hypothesis, I used ABABABA tests, which test for gene flow between non-sister species across a phylogeny. And I recovered all non-significant results in the pairwise comparisons shown here, meaning that we have no statistical support for gene flow in this group. So at this point, I was kind of stumped because we had confirmed this case of mitonuclear discordance, but we found no evidence for any kind of gene flow, which was by far the most common explanation for mitonuclear discordance that I could find in the literature. But eventually I realized I had not given full consideration to the possibility of totally random missorting or incomplete lineage sorting. And what I'm showing here is a species history in the gray tube and a single tree from a single piece of the genome that matches that history. This is in contrast with incomplete lineage sorting, which just means the random missorting of variation. And incomplete lineage sorting will actually cause us to recover trees like the one shown in green, which doesn't match the species history. And we know that this type of random missorting occurs much more often throughout the genome when speciation is rapid. And based on multiple time calibrated trees, we think that the roughly 100 taxa within this kingfisher genus diversified within just the last 2 to 5 million years, making it one of the most rapid avian radiations on Earth. So we should actually be expecting a large amount of this random missorting across the genome in these kingfishers because this rapid branching leaves little time for accurate sorting between speciation events. And I wanted to dig deeper and quantify the exact frequency of accurate sorting according to the species tree versus missorting across the entire genome. So to do that, I decomposed the internal branches on this species tree into rooted three-tip topologies like the ones shown on the screen. And these three-tip trees are useful because they can only take on 3 possible topologies, which simplifies the possible support values, meaning that the minimum support for the most common tree must be at least 33%. And I found that overall, the support values across the tree were close to this minimum value of 33%, which suggests that there's a lot of conflict and random missorting happening throughout the genome. And this is a key insight because the mitochondrial genome is non-recombining and acts like a single piece of the genome. So this random missorting, if this random missorting is pervasive, the mitochondrial genome could be subject to random missorting as well. And in order to quantify the probability of this happening, I was able to use coalescent theory to calculate the probability of the mitochondrial genome sorting accurately according to this species tree. And I found that there was actually a less than 50% chance of the mitochondrial tree sorting accurately for this group at every single bifurcation, given how rapidly speciation occurred. And this suggests that the explosive speciation in this group means we should have expected the mitochondrial tree to differ from the more accurate nuclear species tree more times than not. So finally, we're able to come back to this diagram and say that we think our genomic reconstruction, which is derived from a consensus of thousands of independent pieces of DNA throughout the genome, represents the true evolutionary history of this group, while the mitochondrial genome has actually sorted inaccurately, generating a conflicting phylogenetic reconstruction. So we can move on to the final aspect of this project, taking a step back and asking what this specific study of kingfishers can tell us about how evolution works across the globe. And a main takeaway from this project is that rapid and repeated branching creates an expectation of mitochondrial missorting, which I think is a really important and underappreciated phenomenon in evolutionary biology. Because just as an example, looking at this massive phylogeny showing a rapid butterfly radiation, if mitonuclear discordance popped up, it's likely the authors would have looked through the literature and assumed, like I did, that the explanation must be gene flow and introgression, when in reality, we shouldn't expect the mitochondrial genome, which is essentially just a single locus, to track every single branching event perfectly with no missorting. And it's important to recognize that this expectation of missorting holds especially true when the waiting time between speciation events is short, as is often the case in many of the adaptive and geographic radiations that we're so interested in studying as evolutionary biologists. The main issue is that proving random missorting occurred can be very challenging, so you rarely see it discussed as a viable explanation for mitonuclear discordance in the literature. And going all the way back to the title of this talk, I think a clear takeaway from this chapter is that although there's a single evolutionary history of life on Earth, there is no single tree that can connect all of the extant organisms on Earth in a unified way. And this is because the genetic material within organisms is not inherited in a single uniform block. And this means that rather than searching for a single unified tree of life, we really need to be expecting multiple trees of life that account for the complexity of genetic inheritance. And this brings me to chapter 2, where I'm going to tell you about flower peckers, which are another group of Pacific birds with, I think, equally complex and interesting evolutionary histories. And in this chapter, I'll be talking about the buzzing flower pecker, who gets its name from pecking flowers to get at the nectar inside. And these birds are native to the Philippines. I've outlined the Philippines here on the map with a red box showing its position just off the Pacific coast of Asia. And as I mentioned in the introduction, this is another case where I'm following up on a previous study from the Moyle lab, specifically Hosner et al. 2018, who showed that this species actually contains 3 discrete mitochondrial lineages, which correspond to 3 different phenotypes shown on the right. And I noticed this internal branch on the mitochondrial tree was very short, suggesting rapid divergence, just like we saw with the kingfishers in chapter 1. So I wanted to follow up using genome-wide data to see what it might reveal. So for this project, we're going to build up a brand new conceptual diagram, starting again with our DNA sequence data. And I used the same reduced representation approach for this chapter that I outlined in the previous chapter, this time to sequence thousands of loci from 60 samples spread across the entire area of the Philippines, which are shown by the dots on this map. And I ended up with about 2,500 high-quality loci, or chunks of the genome, with only a small amount of overall missing data. And I wanted to tackle the basic question of whether these 3 mitochondrial lineages found in the previous paper correspond to 3 genomic lineages. So just like in chapter 1, we reconstructed a phylogenetic network, which revealed that there were indeed 3 major genomic lineages that match the mitochondrial breaks, corresponding with a northern geographic group with that olive plumage, a southern group with this highly contrasting black and white plumage, and a central group with intermediate plumage. And right away, the intermediate location and plumage patterns of the central group made us wonder whether this lineage might be experiencing gene flow with the northern or southern lineages. And sure enough, we see these samples from the central lineage that are sort of climbing up the branch towards the southern clade, which is suggesting intermediate ancestry. So to test whether these birds had admixed genetic backgrounds, we used the program admixture to assign genomic ancestry proportions to each sample. We confirmed that the 3 clades in the phylogenetic network were assigned to 3 separate genomic backgrounds, and that those interesting samples from the southwestern edge of the central clade were indeed assigned admixed genomic backgrounds. And this is highly important because this is our first peek into the evolutionary history of the group, and we've now confirmed that there's ongoing gene flow between the central and the southern clades. So now that we understand the geographic distribution of the 3 genomic lineages, we're ready to use phylogenetics to understand their evolutionary history. And to do this, we're going to follow the same approach from the first chapter by inferring a separate tree for each of those 2,500 pieces of the genome, and then generate a single high-level summary of the evolutionary relationships between the 3 lineages I've outlined here. And this is the resulting tree, with the northern group at the top recovered as first branching, or most distantly related. Meanwhile, the central and southern groups are recovered as sister lineages. So this might seem like a very clean story, but 2 things stood out to me right away. First, the relatively low posterior probability value, which suggests that we don't really have high statistical confidence in this tree. And second, the really short length of this internal branch, which suggests, just like we saw in the first chapter, rapid branching occurred between these lineages, likely leaving very little time for the accurate sorting of genetic variation. So just like in chapter 1, I wanted to quantify the exact patterns of relatedness across the genome using rooted 3-tip trees, which makes a lot of sense here because we only have 3 tips total. And what we found right away was that the species tree topology with the northern group as first branching was only supported by about 35% of the genome-wide gene trees. Meanwhile, an alternate topology where the southern group was first branching was supported by about 33% of the genome. And the last possible gene tree topology, where the central group is first branching, was supported by about 31% of genome-wide gene trees. So these gene tree frequencies are extremely close to the minimum support scenario that I outlined earlier, where all three topologies are supported by exactly 1/3 of the genome. And we can actually perform a statistical test here to test the null hypothesis that the true genome-wide frequencies are equal to this no signal scenario, and it's known as the polytomy test. And what we find is that our empirically observed gene tree frequencies are not significantly different from this no signal scenario, where all possible topologies are supported at equal frequencies. So this means that even though the topology we recovered in our species tree is most common throughout the genome, it's not common enough to be considered statistically significant. Therefore, the null hypothesis that these three lineages all branched instantaneously, forming a polytomy or multifurcation and not a bifurcating phylogenetic tree, cannot be rejected. And one additional thing I noticed about these gene tree frequencies was that despite their lack of overall signal, the two alternate topologies were recovered at unequal frequencies. And this is important because under a null model of only incomplete lineage sorting, the two alternate topologies should occur at the same frequency. And deviation from that expectation suggests that gene flow could be distorting patterns of relatedness that we see throughout the genome. And sure enough, when I did an ABA-BABA test, we find one significant signal of gene flow in our pairwise comparisons, this time between the central and northern taxa. So all of these various investigations have now provided us with evidence that the best explanation for the evolutionary history of these three lineages is a polytomy or instantaneous branching. With additional evidence from our admixture analysis at the beginning of the talk for gene flow between the southern and central lineages, and evidence from ABA-BABA tests for gene flow between the northern and central lineages. And this is a very cool and exciting story that sort of summarizes all of the tests we've run so far. But we also wanted to see if we could test support for this specific history in a single coherent framework. And to do so, we turned to a simulation approach. And what I mean by that is that we simulated 4 separate evolutionary scenarios that could plausibly explain the evolutionary history of these 3 lineages. And the full process is quite complex and replicated, but essentially we used a machine learning approach, training a set of 500 neural networks on thousands of individual replicates of each of these simulations. And we then used those neural networks as classifiers and asked them to predict which of the 4 simulated scenarios was most likely to have generated our empirical data. And what we found is that over 60% of these neural networks predicted that a polytomy with gene flow at both areas of contact throughout the archipelago was the most likely scenario, validating the results from all of the analyses that I outlined earlier in a single coherent simulation framework. So now that we have a better understanding of the evolutionary history of these 3 lineages, we can finally ask the key question, which is, what can studying the buzzing flowerpecker teach us about evolution and the search for a cohesive tree of life? And it would be easy to dismiss the importance of resolving the exact branching order between these 3 lineages of birds found in the Philippines, but I believe these results actually have big implications for how we interpret phylogenetic reconstructions and generally understand evolutionary history. And that's because the types of complex diversification histories that we documented here are almost certainly present elsewhere across the tree of life. And as an example, I'm showing you a portion of the avian tree of life here. And when we zoom in on the tree, there are lots of examples of these incredibly short internal branches, just like the ones that we recovered among the flowerpeckers, which suggest a high chance of complex histories, including polytomies and gene flow events. Yet, in higher level phylogenetics, we typically force all of the relationships on the tree to follow a strictly bifurcating pattern that can't represent these types of complex scenarios. And I think the saying, if all you have is a hammer, then everything looks like a nail, applies here. And that's because the bifurcating phylogeny is essentially our only tool for reconstructing evolutionary relatedness at large scales. And it's the only hammer we have in this case. And even though there's a growing recognition of the ubiquity of gene flow at shallow phylogenetic scales, it's often ignored at higher levels. And the main reason for that is that we don't currently have computationally tractable tools that can accurately estimate the complex multifurcations and reticulations that undoubtedly occurred deep in the evolutionary past. And that's okay. This is an active area of development and presents a really challenging computational problem to tackle. But in the meantime, we should be cautious of our interpretations of large-scale phylogenetic reconstructions and remember that they force complex evolutionary histories into these bifurcating binaries, typically without any sort of formal testing for whether this assumption of bifurcation is valid. So in terms of updating the tree of life, I think these flower peckers bring up a major philosophical question in the field of evolutionary biology, which is whether the tree of life is really a tree at all. And I think that this study, along with others, tons of other studies at shallow phylogenetic scales, demonstrates that the true tree of life is unimaginably complex, filled with multifurcations and reticulations. And moving forward, I think there really needs to be an increased appreciation for the true complexity of evolution in deep time, and that we all ought to begin thinking of the tree of life more accurately as a tangled network rather than falling back on these convenient but often oversimplifying bifurcating trees. So this brings me to chapter 3, where I studied a group of birds known as leaf warblers from the genus Spiloscopus. And these birds are montane specialists that can be found in high elevation habitats from Asia out into the Pacific. But today I'm going to be focusing on the region I've highlighted in the map called Northern Melanesia, which includes New Guinea and the Solomon Islands. And zooming in on this map, I'm showing here the regions where Phylloscopus lineages are present as these dark gray shaded areas, which are essentially just high elevation mountaintops on each island. And all of these leaf warblers are small birds that glean insects from the foliage at the top of the canopy, except for on this tiny island called Columbangra, which is an extinct volcano that actually just has a tiny stand of montane forest around the crater rim. And I've outlined this orange area on the blown up map, showing that this montane forest is just 21 square kilometers in total area, incredibly small compared to the thousands of square kilometers found elsewhere across the region. And just to give you a mental image of this extinct volcano mountaintop, these are actually photos that my lab mate Luke DeSico, who spent time on the peak of Columbangra and collected some of the most crucial specimens for this project, took. And these photos show a large portion of this crater rim, just to give you an idea of how restricted this habitat actually is. But despite having just 21 square kilometers in montane forest, Columbangra is actually home to 2 separate lineages of leaf warblers that live and breed in complete geographic overlap, unlike any other island in the region. And specifically, there's an endemic species on Columbangra that's completely different from all of the other leaf warblers. It has this uniquely dark plumage pattern, and it acts completely different. Rather than gleaning for bugs at the top of the canopy, these birds forage by using their long legs and large feet to crawl around on mossy tree trunks. And these are some more of Luke's photos showing this cloud forest habitat from Columbangra with those thick, mossy tree trunks that this unique lineage has come to specialize on. So this is a pretty surprising situation where the smallest stand of habitat for these birds in the entire region also happens to be the only location where 2 distinct lineages occur side by side. And this surprising pattern has been of great interest to evolutionary biologists for decades because they want to understand how species tend to accumulate over time on islands. And there are essentially just 2 main hypotheses for how this might have occurred. The first being allopatric speciation, meaning that the 2 ecotypes on Columbangra evolved separately on different islands, as shown in this cartoon model. And the second hypothesis is sympatric speciation, where a single ancestral population diverged into 2 ecologically differentiated species while living in complete overlap on the same tiny island. And each of these 2 hypotheses also carries phylogenetic expectations. Specifically, if speciation happened in isolation, then we would expect each of the ecotypes to form a distinct clade, reflecting that they evolved separately in physical isolation. In contrast, if the 2 lineages evolved from a single common ancestor on Columbangra Island, then we would expect to see a tree that looks like this, where the 2 lineages from Columbangra are closely related sister species, despite their highly divergent physical appearances and behaviors. And as I mentioned in the introduction of this talk, I'm building here on a previous project from our lab, DeSico et al. 2024, which showed using a few thousand reduced representation loci from throughout the genome, that the 2 ecologically divergent lineages on the top of Columbangra Island are closely related sister species. But in this project, DeSico also found evidence for mitonuclear discordance, suggesting a complex evolutionary history and potential gene flow events. So I wanted to dig further into this potentially complex evolutionary history using whole genome sequencing this time and a greater number of samples to see if we could resolve these discordant evolutionary histories. And I want to emphasize here the collaborative nature of this work, as Luke was the one who secured both key specimens in the field and loans from other museums, and really designed the sampling for this follow-up project, which is shown here on the map, with those bubbles highlighting the populations we were able to include in our whole genome sequencing efforts. And I want to highlight two samples in particular from Isabel and Malaita Islands, where we were only able to get genetic material because Luke was able to extract DNA from the clippings of dried toe pads from two specimens collected in 1927 and 1930. So despite having no source for high quality DNA in these populations, through the power of preserved museum specimens, we were able to sequence entire genomes from birds that lived close to 100 years ago to help resolve the mysteries of their evolution, which is pretty awesome. So we're going to go back to our tried and true project workflow here. And as I mentioned, we sequenced whole genomes for 18 samples from across the entire region for this project, giving us tons of data to work with. And we wanted to use this massive data set to make sure that the individuals we sequence correspond to separate lineages on each island like we expect. So we again reconstructed a phylogenetic network, this time using over 700,000 SNPs found across all chromosomes except the mitochondria and sex chromosomes. And we found that samples from each island were closely related, forming these discrete groups. And we also recovered two genetically divergent clades representing the two ecotypes found on Columbongra. But we also noticed this one specific individual that was not clustering perfectly with the rest of the leaf gleaning samples from Columbongra and was actually connected to the mossy trunk specialist lineage with an additional network edge. And as you might have guessed, this intermediate placement suggests an admixed genomic background. And when I assigned ancestry proportions to individuals, this is exactly what I found. This admixed sample was assigned around 95% ancestry from the foliage gleaning lineage and around 5% ancestry from that mossy trunk specialist lineage, suggesting a relatively recent history of interbreeding between these two highly divergent ecotypes on Columbongra. So now that we had evidence for the overall genetic structure of the group, plus evidence for gene flow between the two lineages on Columbongra, we wanted to summarize the evolutionary history of the group and answer that original question of whether these two lineages on Columbongra evolved in isolation or side by side on the same island. So we used the same approach that I outlined in previous chapters to construct a consensus species tree. And we recovered a fully supported species tree here, where the two lineages on Columbongra are recovered as sister, matching exactly what DeSico found previously using his reduced representation data set. But because we now have evidence for gene flow in this tree, we wanted to dig deeper. So I ran a series of ABBA tests to test for gene flow across the tree. And what I found was this big block of highly significant comparisons between the foliage gleaning lineage on Columbongra and all of the other foliage gleaning lineages from across the Solomon Islands. So on our species tree, the literal interpretation of this result would be 4 separate gene flow edges, each connecting the Columbongra foliage gleaning lineage to the rest of the foliage gleaners throughout the archipelago. And this was a surprising result because in our phylogenetic network, if you remember, we didn't find evidence for gene flow between foliage gleaners. We found evidence for gene flow between the two ecotypes that live side by side on Columbongra. So I thought there could be a simpler explanation here, one that neatly explained both the ABBA tests and the admixture that we observed in our initial population genetic analyses. And that's that the topology shown on the screen, where the lineages on Columbongra are not actually sister species, and the ecotypes across the archipelago form discrete clades. And what I found was that on this topology, only a single gene flow edge between the two lineages on Columbongra, which matches our results from the phylogenetic network, is required to explain the patterns that we observe. So we think that this is the most parsimonious explanation for all of the patterns that we've seen so far, but this is a really surprising explanation, as there are very few documented cases of this phenomenon where the original species tree topology has essentially been washed away by gene flow throughout the genome. So to further test which of these two possible topologies that we've recovered represents the true bifurcating history of the group versus gene flow, we wanted to turn to the sex chromosomes. And sex chromosomes, for a multitude of reasons, typically experience less introgression than other chromosomes, and therefore should be enriched for topologies supporting the true branching history of the group. And I want to emphasize that this is a phenomenon that's been repeatedly validated in birds and other taxa using genomic data. And in this very system, where we see that the gene flow we observed in our autosomal SNP dataset actually completely disappears when we rerun these population genetic analyses using a SNP dataset derived only from the Z chromosome. So to compare these two regions, the autosomes and the sex chromosomes, in a phylogenetic context, we chopped up each of them into 100,000 base pair chunks, made a separate tree from each of those chunks, and then quantified the proportion of the resulting trees that supported each of the two topologies. And as you can see plotted here are roughly 8,000 autosomal gene trees that we recovered. We found that about 68% of the autosomal gene trees support a sister relationship between the two lineages on Columbangra, matching the species tree. Meanwhile, just 9% of autosomal gene trees support a monophyletic relationship between all of the foliage gleaning samples, which was the second topology that I showed earlier. So now that we'd established these autosomal frequencies, we wanted to compare them with the frequencies found on the Z chromosome, which should be comparatively enriched for topologies reflecting the true history of this group. On the Z chromosome, we found that samples from Columbangra were recovered as monophyletic in about 71% of trees. Meanwhile, the foliage gleaners were reciprocally monophyletic in 18.2% of gene trees. When we compare the frequencies of the dominant genome-wide topology between the autosomes and the sex chromosomes, we find a non-significant difference in these frequencies. In contrast, we find a highly significant enrichment for the proportion of topologies supporting the reciprocal monophyly of the leaf gleaning and mossy trunk ecotypes on the Z chromosome. And this is a very important result because it suggests that despite not being the dominant genome-wide topology, even on the Z chromosome, this topology more accurately represents the bifurcating evolutionary history of the group. And we can actually further validate this result by using the F4 ratio statistic to quantify the estimated proportion of introgression between the two taxa on Columbangra on a per chromosome basis. And we find, as expected, that the Z chromosome has experienced the least introgression out of all of the chromosomes in the genome, confirming that it should be enriched for topologies supporting the true history. So, zooming all the way back out to our original goal, we can now generate a complete synthesis of the evolutionary history of this group. We think that the species tree resolved here using whole genomes and in multiple previous reduced representation genomic studies is actually an inaccurate representation of the bifurcating evolutionary history of this group. And that's because this sister relationship doesn't actually reflect a recent ancestor that speciated sympatrically on Columbangra. Instead, we think these ecotypes evolved in isolation at different times on different islands. And only after two divergent lineages came into contact on Columbangra Island and experienced a period of extensive genome-wide gene flow did those patterns flip to support the recently diverged sister relationship that we covered today. And as you might remember all the way back from the introduction of this chapter, this allows us to finally reject the sympatric ecological speciation hypothesis, despite the fact that the majority of the genome supports this topology, and instead support the long-held hypothesis that these lineages evolved separately and reached Columbangra Island independently, despite the fact that this signal has been largely eroded throughout the genome. So going all the way back to our project workflow, we finally have a handle on the evolutionary history of these birds, and we want to know what the surprising patterns we've uncovered can teach us about global evolution. And the first conclusion we can draw is that allopatric diversification, or diversification in isolation, is still a rule among vertebrate taxa living on islands. And this has been the predominant model for diversification since Meyer was developing his classic theories on speciation in the 1940s. And coming into this research, we thought these leaf warblers might be an exception, an incredibly rare case of sympatric vertebrate speciation on a single small island. Instead, we uncovered a surprising story of genomic conflict where the true allopatric history was only identifiable through a series of careful and detailed investigations of the patterns across the genome at a fine scale. And that brings me to my second major conclusion from this work, which is that genomes can lie. And we've always thought that more data would get us closer to the truth than phylogenetics. But here we document a case where gene flow has overwritten the original bifurcating history throughout the genome, to the point that the signals we recover are positively misleading. And this means that the more data we collect, the more confident we become in supporting the wrong answer. And obviously, this has big implications for the field of phylogenetics and our search for a tree of life. All commonly used approaches for phylogenetic reconstruction are based on this crucial assumption that the most common genome-wide topology accurately represents the bifurcating evolutionary history. Here we demonstrate that this crucial assumption can be validated, not validated, in naturally occurring groups of birds. Understanding how frequently this violation occurs across the tree of life should be a priority for researchers going forward, because we don't currently have a good understanding of how often our phylogenetic trees can be positively misled in the way that I've shown here. So this brings me all the way back to the conclusion, where I'm going to try to recap the work I've done in this dissertation and summarize its important implications. In chapter 1, I investigated a case of mitonuclear discordance in the Todiramphus kingfishers and built the case that this discordance occurred simply due to the expected process of random missorting rather than any systematic violation of our phylogenetic assumptions. Based on this investigation, I concluded that this type of random missorting is likely rampant across adaptive and geographic gradations and presents a major underappreciated source of mitonuclear discordance across the tree of life. In chapter 2, I investigated a case of rapid branching with little phylogenetic signal among 3 geographically structured lineages in the buzzing flowerpecker. I built the case that the best explanation for the evolutionary history of this group is a polytomy with additional gene flow where the lineages come into geographic contact. And based on this investigation, I concluded that complex diversification histories, including polytomies and gene flow, are likely common across the tree of life, and that the assumption of strict bifurcation leads to oversimplified and overconfident reconstructions of evolutionary history. Finally, in chapter 3, I investigated a case of secondary sympatry among leaf warblers at the top of the volcanic island known as Columbongra, and built the case that the dominant genome-wide pattern of relatedness actually misleads our reconstructions of evolutionary history and the mode of diversification in this group. And based on this investigation, I concluded that the tree of life is likely to misrepresent the bifurcating evolutionary history of groups where non-sister gene flow is rampant. So this might seem like it's all a bit doom and gloom, but I want to emphasize that this is how the scientific process works. Science progresses through the falsification of previously held hypotheses so that newer, more informed hypotheses can take their place. And highlighting the inadequacy of our current approaches to building large-scale phylogenies, or the extreme challenge of understanding when our phylogenetic assumptions have been violated in these groups of birds, doesn't mean that phylogenetics is an invalid approach or not worth doing. In fact, I want to argue the exact opposite, that the phylogenetic tree and its recent extension into complex networks continues to be the most crucial and valuable tool that we have for understanding evolutionary history. And the purpose of this dissertation is not to diminish the value of phylogenetics, but instead to use some awesome groups of birds to rigorously investigate the limits of current phylogenetic approaches so that we can understand which reconstructions we should trust and continue to improve the accuracy of our understanding of global evolution and the tree of life. At least, I think. I have tons of acknowledgements to do. I'm like sure that I'm going to forget a lot of people, but I wanted to start by acknowledging collaborators in all of the places that these samples came from. But none of this would be possible without the collaboration of the local and native people in the regions where we collected these birds. And Western science has had a problem with parachute science in the past, and it's something that we're still continuing to work on. And I think the field is moving in the right direction, but we need to keep building collaborations that are meaningful so that we can do meaningful science that helps everyone involved. I need to acknowledge all of the museums that donated specimens, not donated, shared specimens for this research. They kept their specimens. And Yeah, there's just a huge list of museums essentially across the globe that gave us tissue samples, access to specimens. Without the collaborative nature of the museum community, none of this would have been possible. I want to acknowledge the funding sources that made this research possible. Rob mentioned I got small grants from the American Ornithological Society and Society of Systematic Biologists to make some of this DNA sequencing happen. And Rob's NSF grant on the birds of the Pacific was essentially the catalyst for all of the work that you've seen here today. And I want to specifically acknowledge the KU Madison and Lila Self-Graduate Fellowship. Specifically Madison and Lila Self, who basically donated the entire endowment personally to fund 40 PhD students every year, which is pretty incredible. The professional staff, that was just a joy to work with during my whole time in the program, as well as my cohort, who was a great source of support outside of like, I don't know, your lab community can become very insular, and it's really nice to be able to talk to other grad students who just do something totally different, and you can each complain about all of your own problems and sympathize. And specifically Christine Chan, who became a really close friend through my cohort in the KU self-graduate fellowship, and I really appreciated her support during this PhD process. I want to acknowledge my committee. Jenny, thank you for being an excellent and supportive outside member. I have like I'm just going to bounce around. I put Mark Holder on here, even though he's not on my committee, because he's like my honorary committee member. I asked him tons of questions. I actually I did my RAC meeting year 2, one of those meetings. And my committee was like, you need to take phylogenetic methods. And I was like, no, I'm just going to do a PhD on the population genetics of scrub jays. I'm never going to build any phylogenies. And I took phylogenetic methods, and it like genuinely changed my whole trajectory at KU and the projects that I was interested in. So thank you, Mark. Jamie, I similarly like took your genome biology class and your R class my first 2 years here, which like laid a foundation for me to be able to do everything that I presented here, which wouldn't have been possible without that. Rich running BIMAL every single week like became one of my most important sources of support and intellectual growth at KU. And I'm just like eternally grateful for the way you invested in me, even though I wasn't in your lab in any meaningful way. Town, who's here on Zoom, I believe. I went to Town's lab meetings for like the first 2 years, every single week for the first 2 years that I was here at KU, because I was on fellowship and like had all this research time, but was a brand new grad student and had no idea what to do with it. And Town was just like, come to my lab meetings and work on a project with me, and was just like opened his lab to me in a way that also was basically totally crucial for everything that I ended up being able to accomplish during my PhD. And Rob, my advisor, for basically opening all of these systems to me. As I mentioned, I came into KU being like, I worked on scrub jays as an undergrad. I love jays and New World birds, and I'm, not that interested in Pacific birds. And basically, through Rob sharing these data with me and encouraging me on these projects, I was able to develop a dissertation that I feel like is quintessentially a Moyle Lab dissertation, and I feel very proud of that. Continuing in that tradition. And on that note, I want to acknowledge Moyle Lab members past and present. All of you were a great support throughout the PhD. And I want to specifically acknowledge some of these older lab members who were sort of the first generation of Moyle Lab members that, as I mentioned, like set the groundwork for everything that I did here. On that note, there's like specific people to thank for each chapter. Mike Anderson did this Todirampus project in 2015, collected tons of samples. Like everything that I did in chapter 1 was built on the foundation that he had laid in understanding that system. I wanted to specifically shout out Jenna McCullough, who was like a very close collaborator on chapter 1, helped with writing the paper, helped with bouncing ideas off of. She completed a whole PhD on just Todirampus kingfishers this week, and is a really meaningful collaborator that helped me get through the PhD process. Pete Hosner did all of this Diceum work and a bunch of other work in the Philippines that also has laid the groundwork for like tons of new genomic projects, new grants, stuff like that. Very crucial. And specifically Luke DeSico, who did the reduced representation stuff on Phyloscopus. And yeah, Luke was like the person who took me out birding when I came and interviewed at KU as an undergrad, and basically was like my main source of personal support throughout most of my time in the Moyle lab. And yeah, I mean, my chapter 3 requires samples that he collected, requires samples that he extracted, and none of that would have been possible without him opening that up to me and being willing to do that. So I'm eternally indebted to you. And I want to acknowledge my family, who basically, supported me, even though I was doing this sort of different science path than really anyone else in my family did. And even when I couldn't explain my research to them in any meaningful way that made any sense to them, they supported me still, and that was all I needed. And finally to Maggie. Maggie, a lot of you know, is like an incredible scientific illustrator, one of the best scientists I know. Her moving here to Lawrence about halfway through my PhD was like a huge turning point in my PhD in terms of feeling supported and encouraged. And yeah, you always encouraged me to be as rigorous as possible in my science and to be as good as I can possibly be. And I'm so grateful to you for that. This wouldn't have been the same set of projects without you. And I have a note that you made the flower pecker drawings for chapter 2 that you saw throughout these drawings. So yeah, this dissertation is like partially your dissertation too, and I'm eternally grateful to you. And that's it. Thanks for listening. Happy to take questions.
Question. Can you repeat them?
Yeah, I'll do my best to remember. Jay?
So your ILS research for your chapter, I really like the research.
Thank you.
But, so one of the things about I think birds have very similar genetics.
Yes. Highly syntenic.
Yeah, and people say how one of the things like your chapter 3 on the C chromosome stuff, specific regions of the G chromosome the ILS should be sorting faster than the other. So if you look at these specific regions of the genome, see whether you can distinguish regions that are now lineage sorted so we get a better understanding.
Yeah, this is a great question. Jay asked if we've searched for specific patterns within chromosomes where regions are more sorted than others. This is literally the only secondary slide that I have of stuff that I didn't put in the talk. This is like where we're trying to go with, especially this third chapter, is looking at the relationship between lineage sorting and recombination rate across the genome. And I basically only had time to plot chromosome 1 really roughly. They're not even perfectly lined up, but there's this exciting pattern where it looks like potentially in the centromere where there's really reduced recombination, we're seeing enrichment for the true history, which is exactly what we expect. So I can't like vouch for any of these patterns across the genome, but I think you're going in the right direction and we're excited to keep following it. Did I repeat the question? Okay.
But for this group of birds, one of the latter phylogenies you have had, one with the white branch being so really long.
Right.
I wonder to comment on that difference in branch length.
Yeah, that's also another thing that we've thought a lot about because the mitochondrial phylogenies, there's like been multiple mitochondrial phylogenies. It's these, sorry, I should repeat the question. Jamie asked, what about the long branch in Diceum leading to the southern black and white group on our phylogeny? And there's been multiple mitochondrial phylogenies published for this group, and they recovered different relationships. And we basically did a bunch of testing and figured out if you force the tree to be ultrametric, that it will kick that group on the long branch out and call it sister. But if you just use like IQ tree and let it do a normal reconstruction, then it recovers a tree that matches the species tree. So basically, we think there's a bottleneck in that southwestern lineage, and there's been a higher rate of evolution in that lineage. Doesn't make it the outgroup to the group, but it complicates everything in both the mitochondrial and the species trees. Yeah, Jenny?
I was gonna say this for later, but I think this is might be good. In the toadheads,
Mhm.
did you modify damage? I'm really interested in.
Yeah, I wish I could say that I did. I did not quantify damage. I talked to Kevin about you. Jenny wanted to know if we quantified damage in the topaz samples. I talked to Kevin Chovanec about using map damage, and he was like, "This is the state of the art." And it's like right before I was turning in my written document, so I was like, "That's going to be a after defense thing." But I'm really interested in too. I mean, you can see in the reconstructions. I think I have them right here. It's these two samples. So they're on long branches. There's dirtiness and messiness in there. And I need to figure out a really good, rigorous way to account for that and make sure that we've handled that.
Yeah.
Thanks. Marlon. Right.
Probably not a tree, but probably it's a network of interaction. But the thing are going to be the consequences of all the other things that we rely on. In terms of biological constructions.
Right.
Association.
Yeah, Marlon wants to know what is the implication of moving from a bifurcating tree to phylogenetic networks if you're trying to do things like map characters onto a tree. This is a conversation I literally just had with Jenna like a week ago, because her dissertation is on Todirampus, and she kept sending me like these totally discordant trees and being like, I don't know what to do. And I'm like, it's just not a single tree, like just accept that. And she was like, but my chapter 2 is mapping traits onto it. I have to have a tree. So this is, I don't have an answer to your question, but like, I think all of us who are working in genomics are like dealing with this in real time. It's like, how can we build more robust models that accurately represent evolutionary history and allow us to do the stuff that we can do on a simplified bifurcating tree? So you should develop models to do it, is what I'm trying to say. Anyone else?
Maybe you'll stick around and.
Thanks for being here, everyone in Zoom. Appreciate it.